Welcome back, everyone. It's Dr. Lindner. Now that we've finished talking about the neuron, right? We went over the structural classification. Is it bipolar? Is it multipolar? Is it unipolar? We talked about the functional classification, whether it's a sensory neuron, a motor neuron, or an interneuron and association neuron. We talked about the cell bodies, the dendrites, the axons, the myelin, the Schwann cell, the node of Ranvier, the terminal, the synaptic end bulbs, the neurotransmitters, right? Lots of content there, and just important to kind of review those things. If you're not memorizing them now, that's okay. It's very rare that students hear it and memorize it. The key is really just hearing it and having an understanding of it. Then you write your notes and you study it and you take it in and digest it a little further. So now we can talk about the neuroglial cells or neuroglial, which means nerve glue. That's what glial means. It means glue, nerve glue. It helps to hold everything together. So neuroglial cells make up about half the volume of the central neural system. They're much smaller than neurons and they're 50 times more numerous. These cells can divide, and when they do, they can create different types of tumors known as gliomas. Now, there are four types of neuroglial cells in the central neural system, and there's only two types in the peripheral neural system. We're only really going to focus on one of those two. So we have astrocytes, oligodendrocytes, microglial cells, and ependymal cells. Those are in the CNS. And I'm going to circle oligodendrocytes here because those are very, very similar in terms of function as the Schwann cells. We know what the Schwann cell does, so then we know what the oligodendrocyte does, except it's real estate, location, location, location. Where is it going to do what it does? So here we're looking at different parts of the neural system. We're going to see a bunch of neuroglial cells. Right here, we have piamata. And piamata is one of the layers that protect your brain and spinal cord called the meninges. We have a piamata, we have an arachnoid, we have a duramata. I call it PAD, PAD. Piamata, arachnoid, and duramata are the three main layers of meninges. So when someone has meningitis, it's inflammation of these layers. Down at the other end, down here at the bottom, we have ependymal cells. Okay, that's a type of neuroglial cell. We have astrocytes. That's a neuroglial cell. We have oligodendrocytes here. We have microglial cells, more astrocytes. Off these ependymal cells, we have cilia and microvilli. We have neurons. There's the infamous neuron. We could see it looks like it's myelinated. You can see the node of Ranvier there. Let's look at some of these functions of some of these cells, especially these astrocytes right here that look like these stars. Notice how they're connecting to blood vessels. That's an important concept for the blood-brain barrier. So the astrocytes, astrology, astronomy, astronaut, right? All dealing with stars, and that's what an astrocyte looks like, a star. It forms the BBB, the blood-brain barrier, by covering the capillaries. And there's a lot of exchange of nutrients. It's involved in metabolizing neurotransmitters. It's involved in regulating potassium balance for nerve conduction and for action potentials. That's the astrocyte. Oligodendrocyte. Remember I said the oligodendrocyte is analogous to the infamous Schwann cell. What did the Schwann cell do? Well, the Schwann cell produced myelin in the peripheral neural system, whereas the oligodendrocyte, that's going to form myelin sheets around axons in the CNS, in the central neural system. Now, this myelin is really important because, again, it's insulating. It's speeding up action potentials. When we have an autoimmune condition against the oligodendrocytes or even the Schwann cells, this autoimmune disease is referred to as MS, multiple sclerosis. MS is when the body is attacking its own proteins or attacking its own Schwann cells or attacking its oligodendrocytes so that the person is not myelinating very well. So action potentials and nerve conduction slow down. Usually the first symptom of MS is going to attack the eyes because the muscles of the eyes are very small and it typically hits the optic nerve, which is cranial nerve number 2. And people will get visual disturbances with MS, issues with the eyes, okay? And it can move down to the legs. The legs feel very heavy, like tree trunks, and um, it could result in paralysis. It could result in lack of usage of the legs, lack of usage of the arms. Person could be very symptomatic in the early stage, and then it can lie dormant for about a decade. For about 10 years, it could be silent. And then some sort of stress can hit the person's life, and then boom, it just accelerates again, and it can accelerate very quickly. So the myelination of neurons, you could see here is the axon, and this is the Schwann cell. And it looks like phagocytosis, right? Like one cell is eating something else, like it's engulfing it. And as it engulfs it, it just kind of makes these circles go, it wraps around, and it keeps wrapping and wrapping and wrapping, producing this myelin sheath. Okay, whether it's the Schwann cell or the axons. Now, microglial cells, microglia or microglial, same thing. These are phagocytic. They engulf and clear away pathogens and dead cells. And they are derived from monocytes. When we look at white blood cells, we have granular or granulocytes, and then we have agranulocytes. All that means is that when we look at a cell that has a nucleus, if we see in the cytoplasm these little grains of sand throughout them, they're called granular sites. And if we see a cell with just the nucleus and the cytoplasm is nice and smooth, they're called agranular sites, A meaning without. So the uh, agranular sites, there are two. There are lymphocytes. Let's go back here. There are lymphocytes, and then we have monocytes. Now, monocytes are always in our blood. They're always in our circulation. They're always roaming around in our blood vessels. And when these white blood cells, these monocytes that are circulating around, when they find a problem in a type of tissue, maybe there's a problem in the skin, maybe there's a problem in the kidneys, maybe there's a problem in the liver. The monocyte leaves the blood vessel and goes into the tissue, and it evolves into a macrophage. And there's different types of macrophages. They just change names. If it's in the liver, it's called a Kupffer cell. If it's engulfing bad cholesterol, it's called a foam cell. If it's in the brain and the nervous system, it's called a microglial cell. All right, so monocytes become macrophages, and a microglial cell is a type of macrophage. Ependymal cells, which we saw here in this picture right there, these ependymal cells, these line the ventricles, and there are four ventricles within the brain. Two of them are called lateral ventricles. We'll put a B for ventricle, lateral ventricles. That's one and two. And then we have ventricles three and four that we'll talk about. So ependymal cells line these ventricles, which are these chambers. Think of it like that. Deep in the brain, we have one, two, three, four. So we have lateral ventricles, one, two, three, four. And then we have, let's say, the sacrum down here. The cerebral spinal fluid has to flow through all of these chambers. And in those chambers, you have ependymal cells that are producing CSF, which is cerebral spinal fluid. CSF, cerebral spinal fluid. They're deep within the brain, and I'll show you where they are in a minute, in a few minutes. Satellite cells, these are just supporting neurons in the peripheral neural system. Pretty general in function. Schwann cell, that's producing myelin. That is going to insulate the axon, speed up the action potentials and the nerve conduction. When it demyelinates as a result of an autoimmune condition, we said that was called multiple sclerosis. Now, there are also certain medications that are called statin drugs, which are cholesterol lowering medications. And statin drugs are known to create neuropathies. When you are unable to produce enough cholesterol, that means it's going to inhibit your myelin production. So you have to be very careful with statins. And remember, your brain is mostly cholesterol. So when people use like simvastatin, Lipitor, Crestor, these are statin medications, they can often result in something called cognitive decline. Some call it dementia. So you have to look at, is some of the dementia in Alzheimer's a result of the medications they're taking and how long they've been on them. So keep that in mind. Body needs cholesterol. Okay. Typically, when cholesterol is going up and cholesterol is produced by your liver, it's usually being produced by your liver to put out a fire somewhere. The fire is typically somewhere in your blood vessels. And if there is damage to a blood vessel, maybe due to high blood pressure, and the epithelial here gets damaged, your body produces cholesterol to patch up the damage. Okay. So cholesterol is a good thing that your body is trying to do to fix damage in your blood vessels. It's just when your body is producing this good cholesterol, if you're incorporating poor lifestyle, like smoking, fried foods, high sugar foods, intense exercise, those things can damage the good cholesterol that you're producing. And that good cholesterol, which is, remember, we have HDLs and LDLs. The LDL gets a bad rap, although it's necessary. When the LDL becomes oxidized, this is the good cholesterol that becomes bad. It has a bad reputation because the LDLs are now damaged, they're oxidized, and that's what's involved in the clotting mechanism of atherosclerosis. The HDLs have a good rap because they bring LDLs back to the liver. But when they're oxidized, we have macrophages that engulf them and they start to occupy and narrow the lumen space. So if blood can't flow through this lumen very well, it becomes very turbulent. And what's important to know when I do my lecture on blood vessels, you will learn in more detail that cholesterol accumulation doesn't happen on the inside of the blood vessels like people think. This is not how it happens. It happens on the outside, squeezing inward. Okay? When we look at the spinal cord on the left, right here, remember the gray matter is the butterfly or the letter H, and the white matter is on the outside. So this is all white matter, and on the inside is gray matter. And then back here is the dorsal horns, and in the front, ventral horns. And in the white matter, this is where we have the white matter is where we have all of the tracks, all of your sensory tracks and all of your motor tracks are all located out here in the white matter. So the white matter is myelinated and the gray matter is unmyelinated. This is the spinal cord. And look what happens in the brain. In the brain, it's the flip-flop of this. In the brain, the outside is gray and the inside is white. Okay, you can see right here, it does a flip-flop. Okay, so right here, when we look, this is the vertebral body. This is your spinous process of the vertebra. Here's your TPs, your transverse process. The spinal cord here is in the middle. So what's the highlighted orange part? Well, that's the gray matter. What's the highlighted part around the gray matter? Well, that's white matter. And let's go a step further. Let's see. What would this be right here? That. So this, since this is the spinous process, this is the vertebral body, so this is anterior, and everything back here is posterior. This neuron, that is your dorsal nerve root or your dorsal root. This is your ventral root. This is your spinal nerve. So your spinal nerve has sensory and motor neurons. Sensory and motor neurons. Okay, when we come back, we will talk about the brain.